Major funding for the 2002 University of Washington Science Forum, Exploring Our World Large and Small, is brought to you by the University of Washington, Office of the Provost, College of Arts and Sciences, College of Engineering, School of Medicine, and the UW Alumni Association. With major funding from WRF Capital, the 2002 UW Science Forum is part of WRF Capital's 20th Anniversary Celebration Series. Additional support is provided by University Bookstore, Millstone Coffee, Dilettante Chocolates, and the University Inn.
Thank you all for taking the time, especially out of your busy work week, to come to the university to learn more about autism. Tonight, I hope to leave you with a better understanding of what we know about the cause, the brain basis, and the treatment of this puzzling disorder. Now, the science that's required to understand something as complex as autism requires many different disciplines to come together and integrate the information across all of these disciplines. Each one of us brings a different perspective, a different technology, even a different language. And the challenge that is inherent in this need to integrate information across disciplines is one of the things that is most difficult about this task. It's also one of the most things that's exciting about it. In fact, at the University of Washington, we now have over 35 MD and PhD level scientists who are working together to try to understand autism, and scores of staff, graduate students, and undergraduates who are working on this problem. So I'd like to acknowledge my colleagues here in this effort. And I'd also like to acknowledge the tremendous support that we've received from Michael Gralnick, the director of the Center on Human Development and Disability, where many of these studies take place. Now, the primary goal of this research program is to try to prevent, treat, and possibly someday even cure autism. However, along the way, we also learn something about normal brain development. We come to discover what a remarkable learning device the human brain is. And we marvel at how easy it is for even a very young infant to be able to interpret a facial expression, to absorb language, to learn to relate to other children. And we see how difficult even the simplest of these tasks are for the child with autism. Now, I'd like to begin by briefly describing how we diagnose autism. And there's impairments in 3 domains. And the first domain is social interaction. Now, this has to do with difficulties in eye contact, in responding emotionally to other people in a normal way, in developing friendships. The second domain is that of language and communication. And here we're talking not only about the use of spoken word, but also the ability to communicate through nonverbal language, to be able to interpret facial expressions, body language, and emotional cues. And third, people with autism tend to engage in repetitive behaviors, and this can be manifested in a lot of different ways. It could be head banging, hand flapping, sometimes ritualistic motor behaviors, and often have preoccupations on certain topics or objects. Now, what I'd like to do is to put a human face to all these words, and I'd like to thank the parents who've agreed to let me show you this little video clip so you can see what a child with autism looks like.
We're planning up to 40 hours a week of home-based therapy.
Right now we do about 15 hours of the one-on-one with the tutor, 3 hours here, and on top of that we do the occupational therapy, physical therapy once a week for an hour. We do speech therapy for 2 hours.
Then he gets tired and he sort of withdraws a little bit and he does his own thing and he's done his homework and he's darn well going to be in his own world right now and that's it, right? And that's sort of what he's doing now, I think. He's tipped off.
Now, the range of cognitive ability that we see associated with autism can often be in the mentally retarded range, but many people with autism are also intellectually gifted or have average IQs. Now, so far, I've been focusing mainly on the impairments associated with autism, and I think it's also important to point out that many people with autism have remarkable gifts, and this can be in the area of music, art, mathematics. And let me just show you some of the beautiful art that has been created by people with autism. This is an illustration done by a local artist here in the Seattle community. Now, one of the startling facts is that the prevalence of autism is dramatically increasing. In fact, when I first started out in this field, this was 20 years ago, the prevalence estimates were about 4 to 5 per 10,000, whereas now our best estimates for classical autism are about 1 in 1,000. For the broader autistic spectrum, 1 in 500, or maybe even more frequent than that. So it's higher than deafness, than Down syndrome, than childhood cancer. No longer can we think of autism as a rare disorder. Now, is this a true increase in prevalence, or are we just getting better at diagnosis? Well, we clearly are getting better at diagnosis, especially children with mild impairments or children at younger ages. But the fact is we don't know the answer to that question, and the reason for that is that there simply have not been good epidemiological studies that have been conducted until very recently. The Center on Disease Control finally is starting to conduct studies where we are tracking what the prevalence of this disorder is. But certainly, this increased prevalence has led to a lot of media attention. And here we have a cover story in Newsweek, which was published 2 years ago. And then just 2 weeks ago, not to be outdone by Newsweek, Time also published a cover story on autism. So what do we know about the cause of autism? Well, we know that genes play an important role. And the best evidence of this comes from studies of twins. And what I have graphed here is the concordance rates. That is, if one child has autism, does the other child also have autism? For identical twins, these are twins that share the same genetic makeup, as compared to fraternal twins, where the twins, this would be like siblings. They share about half of the genetic makeup. And you can see this really big discrepancy between identical and fraternal twins in concordance rates. Now, if we increase the definition to include not just classical autism, but also the autistic spectrum disorders, what we find is that the concordance rate increases to about 70% in identical twins. And if we expand the definition even higher, so now we're considering not full-blown autism, but whether the twin has a social and or language impairment, we now see that identical twins have a concordance rate about 90%, as compared to fraternal twins about 15 to 20%. Now, these rates apply to what we call idiopathic autism. In other words, there's no known genetic cause for these cases in terms of identifying a specific gene. Now, for cases of idiopathic autism, this means that if they have one child with autism in the family, the chances of having a second child with autism are about 1 in 20. Now, if we look at the risk rates for second-degree relatives, so that would include aunts and uncles or cousins, we see a rapid fall-off in risk rates, about 1%. Now, the fact that we see this rapid fall-off from identical twins to second-degree relatives tells us that there must be multiple genes involved, and they're coming together in an unlucky combination for a given child to cause autism. So let's look at how that might occur. So here we have a hypothetical family where the Father is carrying 3 autism risk genes, but may be completely asymptomatic or may have a component of the syndrome. This father may be shy or may have a history of some kind of language delay. And the mother in this case is carrying 2 autism risk genes, but again, does not have autism. And then in one case, the son has inherited all 5 risk genes and then has reached the threshold where autism occurs, whereas a daughter may only be carrying 2 of those risk genes. Now, our mathematical models suggest that autism involves at least probably 5 genes, but we don't know. It may be as many as 20. And the other possibility is that we're looking at 20 genes, and any 5 of those in combination could result in autism. Now, we also don't believe that genes are the whole story. After all, if you think back on those graphs that I showed you earlier, identical twins versus fraternal twins, only in about 70% of cases were the identical twins concordant for autism spectrum disorder. And in the other cases, they either had a mild difficulty or in some cases no symptom at all. So we think that environment is also playing a role. And there's many conditions where the so-called double hit model, where you have an inheritance of some susceptibility for a disorder, but it requires some second stressor on the nervous system for the disorder to be manifest. And here's a variety of possibilities that have been shown in other disorders to be that second hit that then results in the manifestation of the full-blown syndrome. Well, let's talk about 2 of these that have received a lot of attention recently. The first is the notion of viral infection. There's an interesting observation that about 25% of children with autism develop normally, or apparently normally, until the 2nd year of life, and then they lose their skills and develop autism. Now, this happens to be at about the same time that children receive their vaccinations, particularly the MMR, the mumps, measles, and rubella vaccination. And there's been a lot of concern about whether vaccines may be playing a role. In fact, it's of such concern that vaccine rates are now down by 15% in the United Kingdom, and measles is coming back as a disease. Now, this is a very difficult association to try to prove or disprove. You're trying to prove the null hypothesis, trying to prove that there is not a connection. And one way people have studied this is through epidemiological studies. So, for example, when we introduced vaccines in a country, did you see a rise in the prevalence rates of autism? And these kind of epidemiological studies have not found any evidence so far that there's an association. On the other hand, there's some interesting biological studies. So, for example, there's a group in England who studied in these children with regressive autism and also children with other developmental disorders who happen to also have inflammatory bowel disease. He did a gut biopsy and found that they had persistent measles virus in their guts. And so this is raising questions, and it's a very serious question from a public health standpoint, because an answer in either direction is very important. We certainly don't want people not getting vaccinated for measles, mumps, and rubella. But is it possible that there might be a minority of children who do have some sort of immune dysfunction that makes it such that getting this kind of vaccine would then be that second hit? Now, there are many studies now going on really around the world that are trying to answer this question. Now, the second one that's received a lot of attention has to do with toxins in our environment. And the one that's received the most attention is thimerosal. It so happened that we used to use a preservative in our vaccines that contained mercury. And the question came up of what does it do to a young developing nervous system? Again, one that might be vulnerable because of risk factors to have exposure to thimerosal early on. Now, there's been enough concern about this that they now have removed thimerosal from vaccines. But this question still remains whether, again, there may be a subgroup of children who are vulnerable. And to my knowledge, there are now 8 different studies going on around the world that are trying to look very carefully at this question about the relationship between thimerosal and autism. But clearly, lots of kids have gotten vaccines and lots of kids have been exposed to thimerosal. So it's still important that we understand what kind of genetic risk factors might be there that would have predisposed this child, if it is the case that there is a relationship, to developing autism. So let's now talk about how do we find genes. And what I want to do is describe to you 3 different approaches that we're using here at the University of Washington. And there's many other approaches as well, but just to give you a flavor for some of the work that we're doing here and some of the approaches that are used. Now, the first kind of study is called a linkage study. And what we do is to assess families that have more than one child with autism. And for this kind of study, you need literally hundreds of families. So we send out teams of scientists and clinicians all over the United States to find families that have more than one child with autism, evaluate the whole family, including both parents and a non-affected sibling, and to draw blood for DNA analysis. We have families from Alaska, Texas, Florida, New York, even as far as Australia, who have come to participate in this study. We now have evaluated over 200 of these families, and we just got funded to collect 250 more, so that we'll have a sample of 450 families by the next several years. Now, the approach here is to use what's called a DNA marker. Now, these are well-characterized sequences of DNA that don't have any particular health manifestations, but they happen to vary in form across individuals. And what the geneticists are doing is to look to see whether that particular form of DNA marker is tracking and only showing up in the affected kids. If it only shows up in the affected kids, what it tells us is that the actual gene is probably somewhere near that marker. So we're really at the point now of saying which chromosome is it at and what area of the chromosome is it at, but we aren't really at the point where we're saying what is the specific gene. Now, this kind of study, because it is so labor-intensive, involves so many families, also requires collaboration. And we're part of a larger NIH Collaborative Program of Excellence in Autism, where all of these universities participate together in these genetic studies. And they collect families, draw DNA, and send them to Jerry Schellenberg and Ellen Weishman and myself here at the University of Washington for analysis. So it's quite a putting together of many different people in their effort to try to do this work. Now, so far there have been several what we call full genome scans doing this kind of linkage analysis. And thus far, the region that has shown the most positive results is this region of chromosome 7. Now, the interesting thing is that if we characterize families not in terms of whether they have autism or not, but rather in terms of whether there's a presence of a language impairment. So in this case, maybe a dad who had a history of language delay would now be coded as positive. Then we see increased signal on this region of chromosome 7. And this has led people to believe that maybe we're picking up here the location of the gene that is involved in the language component of autism. Now, when Jerry Schellenberg here at the University of Washington and Ellen Weishman first began this work, they started with chromosomes 7 and 8. And in the course of doing the data analysis, they made a very interesting discovery. What they found was that many families, certainly not the majority of families, but larger than would be expected in the normal population, had large deletions in segments of their chromosomes. And just show you kind of the size, if there's any geneticists in the audience, you can get a sense of the size of these deletions. Now, it turns out we all have deletions on our chromosomes, that we have environmentally induced mutations, but we also have natural repair mechanisms that are constantly repairing our chromosomes. So this has led Jerry and Ellen to develop a working hypothesis that there's a possibility that autism may have to do with some kind of problem in a genetic repair mechanism. Now, this is a speculative hypothesis at this point, and Ellen and Jerry are vigorously pursuing this by looking at other chromosomes and trying to understand better this novel finding of deletions. And here you can just see where some of these deletions occurred for some of the families. Now, in another approach, and this was being done by Enrique Villacrez and Jerry Schellenberg, it just so happened that they identified a set of identical twins, both of whom had autism, and in this case, they had an unusual chromosomal abnormality. Now, this kind of chromosomal abnormality is not frequent in autism. It's quite rare, but it's cases like this that can lead to major discoveries in trying to locate genes for a disorder. So in this case, what happened was that part of chromosome 20 got attached to chromosome 7. And what happens when this kind of translocation occurs is that at the break point, this gene is disrupted. So we have a gene where there's been a mutation that's occurred, and we have 2 children with autism. So this provides a major clue to perhaps that gene on chromosome 7 that we're looking for, or one of them. And so Enrique and Jerry have now sequenced this gene, and they're looking to see whether mutations in this gene are present in other autism families in our sample. Now, a third approach, and this approach is really quite novel, and the University of Washington, I think, kind of shines in this area, and it has to do with the fact that we have Ellen Weishman here, who is developing the complex mathematics that are required to do what's called quantitative trait locus analysis. Now, the notion here is that susceptibility genes code for proteins that are involved in specific traits rather than full-blown autism. And I think that you've gotten a sense of that from some of the information that I've given you so far. And the notion also is that these traits are not all or none. It's not as if you either have a social impairment or not, but rather, like height or blood pressure, these can be measured on a continuum. So this approach then assumes that susceptibility genes likely have effects on these continuously distributed traits. And the mathematics now are very different in terms of how we do the linkage analysis. This approach has been successfully used recently to establish linkage for reading disability. And let me just show you how it worked. Now, what I've graphed here is the kind of graph that a geneticist enjoys looking at. And what I have here is a chromosome lying on its side, and here are those DNA markers that I mentioned to you. And what we're plotting here is called a LOD score. It's just simply the degree of association between a particular trait and that location on the chromosome. Now, what you see here is that a component process that's very important for learning to read, and that's phonological processing. That's the ability to make sense out of the units of speech, appears to be strongly linked to this region of chromosome 6. Whereas another component of reading ability, the ability to take those units, form words, and recognize words, shows no linkage to chromosome 6. However, if I were to show you a similar graph like this for chromosome 15, what you would see is the reverse case. So what we're seeing here then is the idea that genes are coding for component processes that act like risk factors, and that these come together then to create more and more risk for developing the full-blown syndrome. So right now we are using this approach with our linkage study, and in fact, Ellen has just finished the first analysis of the first 100 families, and I think it's okay to say that our preliminary results are showing positive linkage on chromosome 2. And we're particularly excited about this because there are 2 other groups in the country who have also shown linkage on 2. So we're hopeful that we might be on the right track here. Okay, I'm going to shift topics a little bit here and focus more on what we know about the brain. And I'd like to begin with the autopsy study. So these are studies where we actually look at what's going on at the cellular level in the brain. And these were conducted over 20 years ago, most of these, and they're now being replicated using much more sophisticated technology. But there are 2 findings that stand out. One is that virtually all autopsy studies have shown reduced number of Purkinje cells in the cerebellum. Now, the cerebellum is an area of the brain that participates in almost all the things that we do. It has to do with coordination during language and attention, It's very important for motor control. And the interesting thing about the nature of the abnormalities in Purkinje cells in the cerebellum is that it suggests that things started to go awry prior to 28 to 30 weeks gestation. So it has given us very good evidence that the abnormality in brain development starts really early, even prenatally, before the child is born. Now, the second abnormality that we see in many cases is in the area of what we call the limbic system, particularly the amygdala and the hippocampus. Now, the amygdala is very important for social and emotional processing, and I'll be talking about that some more in a minute. And the hippocampus plays a very important role in learning and memory. So these also give us some hints about why the child with autism has the cognitive and social difficulties that they do. But what do we know, actually, about brain function? What I want to do now is to walk you through a series of brain imaging studies, and I'm going to focus specifically on the social impairment and help you understand why the child with autism has this profound difficulty in relating socially to other people. Now, what we know is that there are several brain regions that are critical for social interaction, and what I have illustrated here is this complex social brain circuitry. And let's just walk through each of these brain regions. The first is this fusiform gyrus, and this is very important for just perceiving faces. In fact, if you have damage to this area of the brain, you lose your ability to recognize another person by their face, whereas your ability to recognize objects and words, process language is fine. A second area is the superior temporal sulcus. This area of the brain is exquisitely sensitive to biological motion, and what I mean by that is detecting eye movements, mouth movements, facial expressions. Then the amygdala pulls all that together and is able to actually recognize emotional expressions. It can detect the difference between a fear face and an angry face and a happy face. And not only does it recognize emotional expression, but it also has to do with your own arousal or emotional response to seeing that other person's emotional expression. So I guess the way to think about this is the fusiform is saying, hey, I recognize that's a face, and oh, it's moving. I see those eyes moving, that mouth moving. Oh my gosh, that face is sad. And then it takes the prefrontal cortex, which develops later, to be able to take this one step further and to say, I wonder why that person is sad. In other words, to be able to speculate about other people's feelings and thoughts and attitudes, to develop what we call a theory of mind. So all of these brain regions have to act in a very coordinated fashion in order for us to interact smoothly and adeptly with other people. In fact, Temple Grandin, who is a very high-functioning woman with autism, a professor, said, you know, I had no idea that other people communicated through subtle eye movements until I happened to read about it in a magazine 5 years ago. And another adult with autism had said, you know, it's as if the whole world knows how to mind read, and I have no idea how to do this. So we use all of this information from the face to interpret the meaning and the attitudes and the feelings of other people. And this ability is critical for social communication. Now, what I'd like to do is to focus on just one of these brain regions. In fact, we're studying all of these brain regions, as are many other people around the world. But I just want to give you a sense of how we try to look to see whether any of these brain regions are actually impaired in autism. And I'm going to only really talk about the simplest one, the fusiform gyrus and face perception. Now, from very early on, humans are naturally interested in other people's faces. It's certainly not something we have to teach babies to do. In fact, young neonates have a preference for tracking any stimulus that has a facial configuration as compared to an equally complex but non-face stimulus. Now, one way of trying to see whether in autism that this face processing system might be disrupted is to use a technique that's called functional magnetic resonance imaging. So this allows us to look at where in the brain is activated when it's processing a specific type of stimulus. It's a measure of the amount of blood flow, which is a good index of how active that part of the brain is. So in a typical fMRI experiment, these are the kind of stimuli that the person sees while they're lying in the scanner, if you can imagine that. And they're asked to make judgments. Are these the same face or not? And are these the same chairs or not? And we measure the degree of activation while you're making the judgments about faces as compared to the degree of activation while you're making judgments about non-face stimuli. And here's what we find for people with autism. Let's start with the normal individual. When the normal individual is making those judgments about faces, this more medial part of the temporal lobe becomes active, whereas making judgments about objects, you can see another part of the brain is used. In fact, it's clear that there are very different brain systems that have evolved for perceiving faces as compared to objects. The individual with autism, when they're perceiving faces, is much more likely then to use this object part of the brain. Now, does this mean that people with autism cannot recognize other people's faces? No, not necessarily. They may be using areas of the brain that are less efficient and using different kinds of strategies. We think that they use more of a piecemeal approach, looking at individual eyebrows and mouth as opposed to this holistic approach. And in some cases, people with autism actually may have frank problems with face recognition. In fact, just anecdotally, we had a man who worked for us for over 10 years. And it just so happened that one day, one of our staff members was on the bus, and here was this man sitting across from her. And out of context, he could not recognize who she was. And we came to realize, even after having worked with him for many years, that he had no ability to recognize us by our face. And in fact, he used our voice. He was really good at voice recognition, but he also had made a point of memorizing all of the jewelry that the women wore and all of the clothing that the men wore. And of course, we thought this was one of his obsessions and preoccupations, and come to find out this was a means for him to compensate for his difficulty in recognizing our faces. Now, fMRI is very good at telling us where things occur in the brain, but it's not very good at telling us how fast the neural speed is, the speed of processing. For that, we have to turn to another technology, and that is electrophysiology, where we're actually measuring the electrical signals from the scalp. This particular method, particularly event-related potentials, or ERP, is exquisitely sensitive to the temporal qualities of neural speed. Now, what we do in an ERP experiment is we show stimuli, the same stimulus and comparative stimuli, repeatedly. And every time that stimulus is shown, we time lock the electrical signal and record it at the period time locked to that stimulus. Then we average it over several trials. And what happens is that the background activity that's not specifically related to that stimulus cancels out, and we're left with the signal that's specific to the stimulus of interest. Now, the nice thing about this technology is that unlike fMRI, we can use it with very young children. Now, to get a child to go into a scanner means that you'd have to have them lie perfectly still for 30 to 40 minutes. And not only can not children with autism do this, but most normally developing children cannot do this either. So the nice thing about this technology, and we call this high-density ERP because of the density of the electrodes on the scalp, is that we can put this net, completely non-invasive, completely painless net of electrodes on a child in under 5 minutes. And it can even be used with young infants to look at brain activity. So it's very good if you're interested in early brain development. Now here we have a young child looking at a face on a monitor while we're measuring brain activity. And it turns out that normal 6-month-olds will show a differential brain response when they see a face that's familiar to them as compared to an unfamiliar face, and a familiar object as compared to an unfamiliar object. So we decided to do this kind of experiment with children with autism. We had mothers come in. We took a digital photo of the mother's face, and we compared that to a digital photo of a stranger's face. And we also had the mother bring in the child's favorite object, not one that was an object of preoccupation, but just one that was of interest to them, and compared it to another object that they had not seen before. And here's what we found. Okay, this is the ERP of young children, so you can see what they look like. And let's start with the object stimuli. You can see here there's a significant difference in the brain response to the familiar object, which is in blue, as compared to this new object. In fact, the brain's saying, hey, look at that new object, it's more interesting. Whereas there was no differential response in the response to mother's face as compared to stranger's face. Now again, does this mean that young children can't recognize their mothers? No. For one thing, remember that we are showing these stimuli for 500 milliseconds. That's a half a second. So what we're looking at is the brain's ability to very rapidly distinguish between a familiar and an unfamiliar face, something that by 6 months of age the brain is easily capable of doing. Also, it may be that children with autism use other cues. They may rely on touch or voice or smell. We really don't know exactly how the children with autism are processing faces of familiar people versus unfamiliar people. Now, by about age 5, if we measure the electrical signal from this part of the brain in the right temporal lobe, a characteristic signal emerges in response to face stimuli. And let me just show you what that looks like. And what I'm first going to do is to show you the electrical signal in response to a non-face stimuli. And these are actual data recorded from adults who are about to undergo surgery. And before you do brain surgery, we often map out the different parts of the brain to make sure that you don't create any damage to areas that are important. So these are actual recordings from the surface of the brain in adults in response to different stimuli. Now here's what the response looks like to this flower. And here's what the response looks like to a face. And you can see this very robust and well-defined peak at about 200 milliseconds. So at about a fifth of a second, the brain is responding by saying, I know that's a face. So we use this to study neural speed processing in adolescents and adults with autism as compared to normal adolescents and adults, measuring EEG from the scalp. And here's what we found. Here are our data from our own my own lab, where we're measuring the ERP from typical adults in response to stimuli such as these. And you can see that nice, robust ERP component to faces. At about here, we got about 170 milliseconds out. People with autism not only show a less robust response, but you can see it's delayed. And in fact, it's delayed by about 20 to 40 milliseconds slower. Now, why is that important? Well, it turns out to process signals from the face depthly, you have to be able to process information extremely rapidly. And even a millisecond delay can greatly impair your ability to track social information. Furthermore, you have to integrate that information online with other kinds of information. So when you're carrying on a conversation with someone online, you're processing all of those facial signals and the auditory linguistic signal and integrating them over time. So any of those systems are sluggish or get out of whack, it creates a lot of havoc and makes it very difficult, we think, for the person with autism. Furthermore, we think this may have therapeutic implications. It turns out that in another disorder, children with language delay, that they also found that the primary impairment was the ability to process the linguistic signal rapidly. And when they used a computer to slow down the auditory linguistic signal, they found that these children with language impairment could not only process the signal, but they were able to make rapid gains in their language acquisition. So we're very interested in the possibility that we can use these findings to develop some therapeutic techniques to help people with autism in their impairment in face processing. Now we can also look at these signals across the entire scalp. to look at the pattern of activation. And again, here's what we find. In typical adolescents and adults, you see this well-replicated finding that in the right occipital temporal region, you see activity in response to faces at about 170 milliseconds. Whereas in the person with autism, we're seeing a failure for the right hemisphere to specialize for processing of faces. And you also see that they're using dramatically more of their brain to try to process that signal. And what we know from many brain imaging experiments is that the more efficiently and the more capable you are of carrying out a particular task, actually the less areas of the brain that you use. You just become more and more efficient in your use of these brain regions. So this suggests that there's abnormal specialization, but also that it requires tremendously more resources for the person with autism to process a face than a typical person. Now the question is, are we detecting here some kind of early genetic abnormality in the face processing system? And this could be the case. After all, as I said early on, you can see even in neonates this preference for tracking faces. So we know that the face processing system emerges really early in life. And so it's possible that what we're finding here is an early behavioral marker that's related to some sort of genetic abnormality. Now, on the other hand, it's possible that the difficulty lies somewhere else. And the difficulty results in the child with autism not being interested in faces. Faces are not rewarding for some reason. Whatever mechanism is in play that naturally draws our attention to faces is simply not working in autism. In this case, what might be going on is that the face processing system is simply not receiving the adequate kind of stimulation during early sensitive periods when we think these systems are coming online. So what we're looking at now at the University of Washington is whether an early intervention that specifically is targeting attention to faces and social interaction, whether children who respond to these interventions then go on to show more normal patterns of brain activity. Now, before I leave the brain imaging studies, I want to tell you about one other kind of study that's going on here at the university, and these are being conducted by Steve Dager and Seth Friedman. And they're using two different techniques. One, MRI, we've talked about, but in this case, they're using it to look at the detailed brain structure. And they're also using a technique called magnetic resonance spectroscopy, which allows us to look at brain chemistry. So here you can see a typical MRI where you can look in detail at both the gray and white matter of the brain. And Steve and Seth are following children longitudinally during early development to track the way the brain is developing. And they can also then look at chemicals, these three chemicals, choline, creatine, and acetyl-aspartate, and that are found specifically in neurons. Now, these chemicals resonate at specific frequencies in response to the magnet. And they provide for us a sensitive measure not only of cellular function, because these occur in the neurons themselves, but also a rough index of the density of the neurons. In fact, this measure is so sensitive that you will see changes in these chemicals in a person who's had the beginning of a stroke before you'll see it on a structural MRI. Now, so let me tell you a little bit about what Seth and Steve are finding using this in a longitudinal study of young children with autism from age 3 to 6. And the first question that they were interested in asking had to do with overall brain size. Because it turns out that one of the most well-replicated neuroanatomical findings in autism is a larger than normal brain. We knew from other studies that by elementary school age that for some reason children with autism have larger than normal brains, but we had no idea when this abnormal growth process began. So Steve and Seth were very interested in whether even at age 3 we would see this larger than normal brain, and indeed that is what they found. They found that children with autism by age 3 have larger brains than typically developing children, and also 3- and 4-year-old children matched on IQ who are developmentally delayed but do not have autism. Now, we know from looking at head circumference at birth that these children probably have normal brain size at birth, because it turns out that head circumference is a pretty good proxy for overall brain size at birth. So there appears to be some kind of postnatal abnormal brain growth process that's occurring that we still have very little understanding of. But one of the hypotheses is that perhaps this is due to an abnormal process of neuronal cell death, and let me explain that. It turns out that in normal brain development, our brains actually overproduce the number of neurons, and then through genetic mechanisms, they naturally die off, and we then have the number of neurons that we have as an adult. Similarly, it overproduces the number of synapses, and through both genetic and experiential mechanisms, these are reduced. So a 1-year-old actually has more synapses than we do. So one of the hypotheses was that perhaps what's going on here is that these children simply did not have the normal process of cell death, and so really what we have is more cells here. Now, if that were true, what you would predict when you look at those brain chemicals is that children with autism should have higher levels of those brain chemicals. But here's what Steve found. He actually found that the children with autism had lower levels of NAA and creatine and choline than children with typical development and delayed development. So we think that the hypothesis that this is due to abnormal cell death is probably not correct. But what is causing this abnormal brain growth? We really don't know, but there are several groups around the country right now who are examining genes that regulate brain growth. And we're very interested in this. If we could discover a gene that specifically relates to the mechanism involved in this brain growth, then theoretically you might be able to screen kids early on and actually intervene in this process. I want to turn now to telling you a little bit about early detection and treatment. Now, 20 years ago, it was very uncommon for a child to be diagnosed with autism under the age of 5. Certainly when I did my training, we never diagnosed a child until they were elementary school age. Whereas today, autism can be detected by 18 months in most cases, and there are good screening batteries that are now being used at 18 months of age. However, unfortunately, the average age of diagnosis is still 4 years of age. Why is this the case? Mostly because health professionals, pediatricians, and other people that are out in the community simply don't have the knowledge that they need to be able to recognize autism early on. People are trained to look for physical development, to some extent language development, but there's very little training on some of these social and emotional cues that are so critical in being able to detect autism early on. So we feel like this is a very important area where there needs to be more education of professionals in the community in recognizing autism. Now, we also think, because of the nature of the difficulties that I've already described to you in autism, that it should be possible to recognize autism in very young infants. Face processing, emotional processing, even a 2-month-old, when they see a still face, will respond to that face by getting distressed. So by 2 months of age, we're able to pick up on the emotional cues in other people's faces. So it should be possible to recognize autism in early infancy. But how would you show that's the case? Well, one approach that we've used is to actually study home videotapes of infants where the infants were videotaped by their parents, and then we can examine these at a later date. And in the first study, and this was done by Julie Osterling and myself in 1994 now, we wanted to show that you could recognize autism at least by 1 year of age. And Julie had the brilliant idea of studying first birthday parties. It's a great idea because you know the children are 1, exactly 1, and also there's something kind of standard about a birthday party. There's the bringing in the cake, and there is the singing and the opening of presents. And the interesting thing is there's an actual experiment in developmental psychology. It's called a social referencing experiment, and it's designed specifically to know whether an infant can pick up on the emotional cues of other people. What you do is you bring in a novel object, a very unusual object the child's never seen before, and if the experimenter gets all excited and happy, then if the baby's picking up on that, the baby will approach and be interested in that. On the other hand, if the experimenter looks fearful and apprehensive, then the babies will tend to withdraw. Well, what better novel object than a flaming cake to bring in and place in front of a young infant? So in fact, what you see in this naturalistic social referencing experiment is that a normal baby looks around to see how all these other people are responding. Of course, everyone's singing and smiling, and so they realize this is indeed a happy event. Whereas the baby with autism is focusing more on the plate and the cake. And we actually found that 4 behaviors distinguished virtually all of the 1-year-olds with autism from virtually all of the typical babies. And those 4 behaviors were: 1, looking at faces; pointing and showing, which are early gestural behaviors. We call these joint attention behaviors, and they're the infant's ability to draw attention from another person. And then orienting when their name was called. And by the way, that's another behavior you get a lot of chance to see, because people run around with their video recorders and say, "Johnny, Johnny." And so you can see whether the child is orienting when they hear their name called. Now, here we happen to have 2 people that are world-renowned experts in infant development who are trying to develop early infant markers for autism, and along with myself. And we believe that these 3 behaviors hold a lot of promise for eventually being able to recognize infants with autism as early as maybe 6 to 8 months of age. The first one is attention to faces, which we've already spoken a lot about. And the second one is attention to speech. And you may recall I mentioned a moment ago that the baby with autism didn't orient when they heard their name called. Now you see babies normally orient when their name called about 7 months of age. Now what Pat Kuhl here at the university, who's a world-renowned expert on the development of speech in infants, she has found that many young children with autism actually have problems in being able to distinguish the phonemic units of speech. So she did an experiment using that electrophysiology technology that I showed you earlier to see whether children with autism had differential brain responses when they heard the sound paw as compared to ba. Now, very young infants will show a differential brain response to those 2 stimuli. And she found that many of the children with autism were not making this simple perceptual distinction between these phonemic units. Now, this tells us that many of these children have fundamental problems in speech processing. And by the way, most people thought that autism did not involve that aspect of language. They thought mostly it had to do with the social and emotional component of language processing. Now, Pat has also found that young children with autism don't show a normal preference for speech sounds over non-speech sounds. So this is also possibly going to be a useful early indicator. And finally, Andy Meltzoff is looking at the process of imitation. And as you may know, babies love to imitate other people. And not only that, imitation is a skill that develops very early on. Even neonates are capable of imitating the facial expressions of other people. And it turns out that children with autism have a profound difficulty in the area of imitation. Some of these children appear not to be able to map their own body movements onto other people's body movements. Other children seem to be pretty good imitators, but they simply don't have the proclivity to imitate in social situations. So we think that imitation also may be a useful early marker. Now, why is it important to identify these kids so early? Well, what we've learned is that early intervention is critically important. And by early intervention, what I mean is the child needs to receive a minimum of about 25 hours of intensive one-on-one teaching, where the most basic skills of making eye contact, sitting in a chair, orienting to speech are taught systematically one by one. And that these interventions go on for 2 to 3 years during early development. And we find that if children are diagnosed by age 2 to 3 and receive these early intensive intervention, that many of these children have very dramatic gains. Now, not all of the children respond dramatically, but a large proportion of children have IQ gains that can be 2 standard deviations, 28 points, so that early on they would be functioning in the mentally retarded range, and by age 5 would have a normal IQ, go into a regular classroom, be able to develop friendships, and go on to live a normal life. So these are very important for children to receive. Now, last year, the National Research Council, this is a group of scientists who advise Congress on social policy, met to make recommendations about what kind of treatment children with autism should be receiving. And they made the recommendation that children with autism should be receiving at least 25 hours of intensive early behavioral intervention during the preschool years. Now, here at the University of Washington, we're very lucky that through a donation, a private donation, we were able to develop an autism center where now we have increased accessibility to these interventions for children with autism in the greater Puget Sound area. And we've also developed a professional training program with the idea of training people in our community to be able to deliver these interventions so that we can increase capacity of these children to receive these interventions. But accessibility is just one step, and the other issue is affordability. So these interventions cost at least $25,000 per year, and at this point, few insurance companies cover it. So parents must either forego these interventions for their children, or they must shoulder the burden of this financial strain themselves. Now, I'm extremely pleased to say that Microsoft Corporation, through parent advocacy in conjunction with people here at the Autism Center, is the first company, to our knowledge, to pay for an insurance benefit coverage of $75,000 for each child with autism to receive this type of intervention. And we applaud Microsoft for doing that, and we hope other companies will follow suit. Now, these kind of interventions obviously make ethical sense. They have profound effects on people's lives. But it turns out that they also make financial sense. There have been at least 2 cost-benefit analyses that have been conducted. One which showed that by 22 years of age, it's a $200,000 cost savings per child, and by 55 years of age, $1 million cost saving per child, because they're not needing to use the mental health and other kinds of services through the schools that are required when they don't receive these interventions. Now, early detection and treatment are clearly important, and this brings us full circle back to the beginning of my talk, when we talked about the importance of discovering genes. Clearly, the goal is to find the genes, and we can imagine the day when we might be able to screen very young infants for susceptibility genes to autism. We might be able to know which child should have this kind of vaccine regime versus that. We might be able to intervene early on. Farther in the future, we can also look towards gene therapy, particularly as we come to better understand how genes regulate brain development. Furthermore, by understanding the gene, we will come to understand the basic biologic defect in autism, and this will immediately point towards drug therapies that can be useful. So in the beginning of my talk, I said I hope to leave you with a better understanding of the cause and the treatment of autism. Tonight, I've only been able to offer partial answers and clues that can guide us on this search. I haven't been able to provide many solid answers to the puzzle of autism. However, I'm very impressed with the advocacy and the dedication of so many families and of the scientists. And along with that, I see amazing advances in the technologies that are being used to study autism. So what I believe I can offer tonight is hope. Hope for new answers that someday will make a real difference in the lives of people with autism. Thank you.